start the video recording, and we are synchronized now. Okay, so we just talked about macroglia of the peripheral nervous system, and now we're going to talk about macroglia of the central nervous system. And we are going to start with the macroglia I mentioned over here, astrocytes. Okay, astrocytes. All right. There we go. Okay, so astrocytes are also called astroglia. And now that we've separated this into two words we are already familiar with, astro and glia, take a guess at what shape this glia has. Okay, but before that, we are going to remark that it is the most abundant macroglia in the central nervous system. So, most abundant macroglia in central nervous system, and it they link neurons to the circulatory system, to the blood supply. Okay, while helping form the blood-brain barrier. Okay, we're going to talk more about this later, but it is a new word, so I'll underline it. Okay, they also are involved in neurotransmission. We've already talked a little bit about that. So they recycle before that, they remove excess potassium ions from around the neurons, okay? Remove excess potassium ions. And remember, an ion is a charged particle. So the chemistry shorthand way to write this is K with a plus, because on the periodic table of elements, the symbol for potassium is K. So they remove excess potassium ions from around neurons. Okay, and why is this important? So you remember when I drew the more realistic scale of our multipolar neuron with the long axon. So the way that electrical signal travels down that long axon, it involves potassium going in and out of the axon membrane at those nodes of Ranvier, the blank spots in between the myelin sheaths. So potassium is moving in and out of the axon membrane. And so if there's too much of it around, it can mess up the electrical signal. So that's why astrocytes are really important, and they are generally found, I haven't said that yet. Yes, so that's why this function is very, very important. Okay, next, they recycle neurotransmitters, which have been released during synaptic transmission. Release neurotransmitters that have been released during synaptic transmission. Okay, lots of new words here. That's fine. We'll underline them in red. And we'll do a quick jump to define them. So, a neurotransmitter is a chemical that is both created and released by a nervous system cell, okay? So both created and released by a nervous system cell. That distinction is important because nervous system cells can also use chemicals that haven't been created by themselves as chemical messengers. But specifically, neurotransmitters are a chemical created and released by the nervous system cell that acts as a chemical messenger. That acts as a chemical messenger. Okay. All right, then synaptic transmission. We're going to give a very brief overview right now because we're going to actually talk about both of these in greater depth later. So, synaptic transmission is when I just have a drawing here. What's going on? Okay, so synaptic transmission is when a bit of chemical, the neurotransmitter, is sent across a tiny gap in between cells called the synapse, okay? Synaptic transmission is when a neurotransmitter, which I'm going to abbreviate as NT, okay? When a neurotransmitter is passed from one nervous system, NS, nervous system cell to another. Okay? So we'll go into more detail about this later. All right. So speaking of neurotransmitters, the neurotransmitters that astrocytes use to communicate with each other, with other astrocytes, are, so they use glutamate, GABA, and ATP to talk to each other. Okay. All right, so lots of new words. Glutamate and GABA we're going to go into in great detail later because they are also the two most common neurotransmitters in the central nervous system. So they're really important. Okay? We're going to go ahead and define ATP now. So ATP stands for adenosine triphosphate. And this is a molecule used for energy by most living cells. Yeah. Molecule for energy for most living cells. Okay. All right, so now let's draw our astrocytes. So there are two structures that astrocytes can have. Let's do this. Okay. Two structures. Okay, so our first one is called protoplasmic. Protoplasmic. Okay? Let me just check that you can see that. It's not great. I will choose a different color. All right, so 2 structures. I think that's better. Okay, 1. Protoplasmic. Okay, and this structure is short, thick, really heavily branched processes. Okay, so what does that look like? We've got short and thick. Heavily branched. Okay. Oh boy, I'm getting carried away. All right, there we go. Short, thick, heavily branched. Whee! All right, protoplasmic astrocyte. And these are mostly found in gray matter. Okay, what is gray matter? Okay, gray matter. Okay, so when you look at a brain under a microscope, usually what has happened is the brain has been frozen either through temperature or the lack of temperature, so you actually literally freeze it, or you use chemicals that chemically freeze it in place. And this is because a brain, like brain tissue at room temperature, if you've done nothing to it, it's like jello and it's going to fall apart and you have to scoop it up with a spoon. So you have to freeze it. And then once you've frozen it, you have to slice it into very, very, very thin pieces, and then you have to dye it. Okay, otherwise it all looks transparent. There's really nothing there that you can see unless you dye it. Okay, so once you've dyed it, people saw under the microscope what looked like white matter, and then some darker parts of the brain, and they called it gray matter. And it turns out later on we found out that the white matter is actually the fatty lipid from the myelin sheaths that we were seeing, and the gray matter is more just a bunch of cell bodies, or not so much the fatty lipids. So that was the big defining feature. Okay, so the white matter is where there are lots and lots of axons, especially long axons that require myelination. Remember the fatty tissue that insulates the axon? Yeah, so protoplasmic is found in the gray matter, which is mostly cell bodies and non-axon parts of neurons. Okay? Then our second structure, shall I do it? Yeah, I've got space. Okay, so we'll do that over here. Number 2, our second structure. This one is called fibrons. Okay, and if the protoplasmic was found in the gray matter, where do you think the fibrons are? They are in the white matter, so found in white matter. Okay, so they are more associated with axons and myelin sheaths. Okay, so let's draw one. All right, so there's gonna be the start of a body, and then it kind of does this long, thin thing. Okay. There's probably a couple more coming off, to be honest. Okay, now it looks like a running glia. All right, so there's our fibron, found in the white matter. They're long, thin, and much less branched in their processes, okay? And that does it for astrocytes. All right, so we have 2 more macroglia of the CNS, but since I've run out of space, I'm going to erase this and give myself some fresh room. Oh man, if we were in real life, you would probably all be yelling at me for erasing too soon, but since it's in a video, you can just pause the video.